Welcome back, ladies and gentlemen. Now we will continue with perhaps the most promising area yet, the cellular therapies, and continue with the presentation. We would like to invite Dr. Hwang Shiaw-Min, CEO of U-Neuron Group, who will introduce breakthrough in cellular therapy. Ladies and gentlemen, please give him a big applause.
Yeah, firstly, I would like to express my sincere gratitude to the organizing committee for the invitation. And today, I'm delighted to share with you our work in Taiwan. And the title is a little bit shift from macro view of cell-based therapy into a more specific of our experience. We use the amniotic fluid stem cell and exosome. I will introduce the new tools in regenerative medicine. And this is my disclosure for your reference. And this is the content. I will mention in the order of the first part, cell therapy. Second, AFSC source. The third, the clinical trial preparation. And the new drug pipelines. And last is the exosome. Okay. As we all know, there are three waves of medical revolution in terms of the pharmaceutical industry. The first revolution we called the small molecular drugs. We use the chemical synthesized compounds like the aspirin, antibiotics as medical tools. And the second wave of the revolution we called the macromolecular drugs or biologics. They will use the protein, polynucleic acid, and the vaccine as the drug. And now we shift to the third revolution. That's we call the cell and gene drug or cell and gene therapy, CGT. We use the live cell as the tools. This is the so-called cell-based therapy, and they can recognize as the next pillar of medicine. So we are transitioning from the sick care, that's the fighting for the pathogens, into the true health care. And we use the live cells to restore the organ function and help people get well. And that's the essence of the regenerative medicine. And about the global market of regenerative medicine, we know that North America is the largest market, but I want you to focus on the central parts. The Asia-Pacific region countries are the fastest growing market in the next few years. And that's according to this market reports. Okay, and for the regenerative medicine, we need the stem cells. And why? And from the amniotic fluid. Amniotic fluid we can get by the aminosynthesis. Amniotic fluid is called 羊水. Aminosynthesis is 羊膜穿刺. Okay, it's a prenatal diagnosis procedure during 16 to 20 weeks of pregnancies. Okay, and this amniotic fluid contains the early stage of fetal stem cells, very close to the embryonic stem cells. And these cells can differentiate into the mesoderm and the ectoderm cells. The ectoderm means the nervous and the skin epidermal system. And because this is the prenatal diagnosis procedures, there's no ethical issue. And the last, but not the least, is the amniotic fluid. There's no blood content at all. As you know, the development history of MSC is from different tissues. The first generation of MSC is from the bone marrow. That's the gold standard of MSC. But the challenge is invasive harvestness and the low frequencies of this source. The second generation is from the adipose tissue. We call it the ADSC or ADMSC. The challenge is the cellular senescence and the cell differentiation. So move to the third generation of MSC from the placenta or cord MSC. Although this is the postnatal 0H, It's recognized as a birth medical waste, but the challenge is the microbial contamination risk. And the fourth generation and the newest generation of the MSC tissues is from the amniotic fluid. Okay, it's a routine prenatal procedure and no ethical issue. And you can see from the generation 1 to the newest generation, the source is from younger and younger, and no ethical problem. And else from the blood-related tissue to the non-blood-related tissue. In the right-hand side, you can see the 1 to 3rd generation, we call it the adult stem cells. Only the amniotic fluid, we call they contain the fetal stem cells. Okay. This is just a picture to illustrate the different tissue sources of MSC. You can see the first bone marrow aspiration for the bone marrow MSC, the liposuction for the ADSC, the placenta and the cord for the birth medical waste, the MSC. Okay, and these are postnatal and the blood related tissue. In the contrast, for the amniosynthesis, okay, under the ultrasound guidance, only 20 ml of amniotic fluid is safely aspirated by amniosynthesis. As you can see here, in the syringe, that's the fresh amniotic fluid. It's clear and transparency fluid, no blood at all. Okay. We are the first group to successfully isolate and characterize the amniotic fluid stem cells. And my paper published in the 2004 in Human Reproduction, and there's a cover feature to introduce our publication. Right now, it's over 950 scientific references. And we have patent secured the AFSC technology. Okay. And 3 years later, in 2007, AFSC or AFMSC has been validated by the JAMA journal. The JAMA recognized the AFC therapeutic potential. And that year, the Time magazine selected the finding of amniotic fluid stem cells as the top 10 medical breakthrough. That's for your references. And I just summarized the advantage of AFMSC compared with other MSC. The first is the functionality. We call AFSC is polypotency, whereas the other MSC are multipotent. And the telomere length is the longest because it's the early development. And you can see the picture here. That's the telomere assays results. And there's no tumorigenic issue. And these cells grow faster and the low immunogenicity and the genetic stability compared to other MSC. And all this evidence led us concluded that AFSC have ideal safety and efficacy profile for regeneration application. And this is the potential of AFC in the clinical application. Our company just focus on the neurological repair, we call it for the stroke, Parkinson, and the neural damage. And the other group will use this AFSC in the heart diseases, kidney diseases, diabetes, or artificial reproduction and the lung fibrosis application. And last but not least is because AFC is you can get actually the youngest cell source that can be used in the anti-aging formula. Okay. And I just mentioned this, this is just published in March this year. A paper compare the AFMSC head-to-head with the cord MSC as a therapeutic tool against the cytokine storm and other inflammatory syndromes. To make the long story short, I just summarized the result. They use the gene set enrichment analysis tools compare head-to-head AFMC over cord blood MSC. In terms of the regulatory pathway, they use the hedgehog, angiogenesis, and notch signaling. And you can see the clear superiority of the AFMSC. And for the SARS-CoV-2 associated cytokine storm pathway, in terms of the acute inflammatory cytokine, like the TNF-alpha, IL-2, IL-6, interferon gamma, all you can see here is the superiority of AFMSC versus cord MSC. Okay. And the conclusion of this paper is the AFMSC have superiority, regenerative, and immunomodulatory capacity against the cytokine storm and the inflammatory syndrome. Okay. And we know although the science is good and it makes sense, but research alone have saved no life. That's why we set up a company to proceed the cell therapy clinical development using AFMSC. Of course, we are based on the science evidence and trust in the cell source. We have the manufacture process and the GMP facility. Also, we need to organize a sophisticated team to follow step-by-step of clinical trial paths and the regulatory paths. Okay. This is our flow chart of the CMC, the chemistry, manufacture, and the control of the AFMSC. Of course, the first one, we have the IRB approval. You do the donor eligibility to donor screening and testing to qualify your donors. Okay. Then collect the amniotic fluid, then send to the GTP certified or GMP compliant facility to isolate these cells and the expansion, make the master cell bank, then make the drug substrates. And under the cryogenic storage material, okay, cryogenic storage, okay, all of this protocol has been approved by Taiwan FDA for 2 protocols indications. And this is just a picture of our cell processing center. As you can see, this is the so-called the GTP certified or GMP compliant, okay, the cell culture room. And the other part is the 24 hours monitoring, okay, 7 days, okay, the cryogenic storage room, okay. One more thing, okay. We will mention about what's the mechanism of your stem cells, and most of us agreed the mechanism of action of stem cells is primarily attributed to paracrine effect, or we call the paracrine signaling. And the key components has been growth factors, or anti-inflammatory cytokines, or the extracellular matrix, or the last, the exosome. And these are all your stem cell produced factors. And that brings us to focus. Exosomes are critical mediators. So I will mention about what is the exosome. The exosome, we say the mechanism of our stem cells. They are generated from intracellular MVB, multivesicular body. And these exosomes are lipid bilayers membrane. And narrow size, of course, they are 30 to 150 nanometer vesicle. And they carry lipid, protein, RNA, especially microRNA, and other bioactive substance as a cargo. And the function of these exosomes are recognized as the cell-to-cell communication, or the messengers. Okay. And this, again, is our flow chart how to produce the exosome from the amniotic fluid stem cells. Of course, we have, again, the IRB approval. That's essential. Do the aminocentesis and the AFSC isolation and expansion. Then we collect the conditioned medium under the GMP compliant or GTP certified facility to do the downstream processing via centrifugation or tangent flow filtration, or called the TFF, then size-sieve the purification. After that, we can do the formulation, and that's our products. To do pass the QC, then you can do the clinical trial. Okay, and this is the other group's publication in the January of this year. They compare amniotic fluid stem cell derived extracellular vesicle. Extracellular vesicle in terms of just the exosome for short. Okay, in this paper, they showed the amniotic fluid stem cells exosome has unique, large quantity of microRNA content compared to others. Okay, compared to other sources of the MSC. Okay, for your reference. And the beauty of this microRNA are measured for the regenerative capacity. That's what it means. As our data, this is our data. We do the RNA sequences, and that's our top 10 microRNA. The first one is we call microRNA 199A. The function you can check. This microRNA is for the myocardial regeneration. And the second one, microRNA 199B, is for the bone regeneration. And the third quantity is for the angiogenesis. The fourth is for the tumor apoptosis. It actually inhibits the tumor formation. That's the microRNA 165B. And the other one, you can follow that. That's for the wound healing, ECM remodeling, neuron repair, something like that. It's all, again, to show our superiority in regeneration capacity of EV. Okay. And this is our COA. Okay. We are, each lot, we have the COA. We call the C miracle, or we call the EV derived from amniotic fluid stem cells. This is our report. In the left one is you will have that. In this COA, you will have also the NTA report, narrow particle tracking analysis report. You can see the flow chart, okay? You can see the exosome size and the concentration report. The most important you need to know is the exosomes marker. As we know, the exosome contains the CD9, CD63, and 81. That's our narrow flow analysis report, and to show the purity. So when you have the COA, you will know the size distribution, the mean or the main size of your exosome, and also, of course, the concentration of your products. Okay. Okay, that's the last. I thank you very much for our slogan is the U-Neuron. It's a promise you a better life. Thank you very much.